Hey guys, today I will be talking to you about proteins. Specifically, we will go through protein digestion, and then we will talk about a process called protein deamination and protein amination. Now, before we can talk about proteins, we have to talk about what proteins are made up of, which are amino acids. There are non-essential amino acids, which are amino acids that the body can make itself. And then there are essential amino acids, which are amino acids that we have to obtain from nutrition, so we have to intake them every day. There is also a group called branched-chain amino acids. These are important because they go straight to the energy train and can become ketones, which is very beneficial to the body. Now that we know what proteins are made up of, we can now talk about the two types of proteins. The first type is called complete protein. This is high quality, and they are foods that provide all the essential amino acids. The second type is incomplete proteins. Now these lack all amino acids and they can't build tissue without help. Now we will be talking about protein digestion. Now when you first eat protein, like that big old meaty steak, you first chew it. However, no digestion really occurs during that phase. It first occurs once you hit the stomach. In the lining of your stomach, you have these things called gastric pits. Now these gastric pits contain many cells which help with the digestion of proteins. Now once proteins hit your stomach in forms of large polypeptides, that then signals G cells to release gastrin. Then gastrin then goes through the capillaries and tells the chief cells to release pepsinogen and the parietal cells to release HCl. Now when these two mix, it forms pepsin. Now pepsin and HCl help break down the polypeptides. As you can see, after digestion in the stomach, the polypeptide chains are now only 4 to 5 chains long. Once the acidic chyme, which contains your proteins, comes from your stomach in the duodenum, your S cells then release a hormone called secretin. Secretin then goes to the pancreas and goes to the duct cells of the pancreas. They then secrete HCO3, which goes back in the duodenum and raises the pH to a pH of 7 through 8. This raised pH then signals the I cells to release CCK. This causes the release of proteases. Now these are inactive forms, but once it reaches the duodenum, trypsinogen then is activated by enzymes, and then it activates all the other enzymes as well. These enzymes then break down proteins even further into tripeptides and dipeptides. Once they are in the jejunum, they go to the brush border. Now there are enzymes waiting for them at the brush border to break them down into single amino acids. These amino acids then go into the liver, where transamination and deamination will occur. The process of transamination is taking an amine group off from a carbon and recycling it. The enzyme transaminase enzyme help break down alanine. This alanine then turns into an alpha-ketoglutarate, which is a keto acid. Carbon skeleton of the alanine is then turned into either ATP, another amino acid, or used for storage such as glycogen or ketones. Deamination is in the process of total catabolism of the amine group in the carbon skeleton. So, once alanine has been broken down, its amine group is then carried off via glutamate, which is the universal carrier for nitrogen. This glutamate then goes through the bloodstream and then goes to the liver. In the liver, the enzyme glutamate dehydrogenase breaks it down into a keto acid and NH3. This NH3 then combines with hydrogen to form ammonia. This ammonia then goes into the mitochondria of the liver to create the amino acid citrulline. It then goes through the urea cycle to create other non-essential amino acids for the body. A byproduct of this is urea. 90% of the time, transamination is happening, and 10% of the time, deamination is happening in a healthy adult. Now, if you're always eating junk food, your body will not go through that normal process, which is not good because it causes an increase of ammonia in the liver. Glutamate synthetase pulls in the extra ammonia into the brain. This ammonia then goes into the astrocytes and combines with glutamate in order to form glutamine. Glutamate creates GABA, so if you have a lot of ammonia, you are not able to create a lot of GABA, which can lead to anxiety. When there is also excess ammonia in glutamate, it also causes a lot of water to come into the brain. Astrocytes are an important part of the brain. When astrocytes get sick, their metabolism decreases, so they use glutamine for energy. They feed glutamine right into the mitochondria to the neuron. If glutamine is broken down in the neuron, it creates ATP. However, a nitrogen problem will occur in the mitochondria of the neuron. This will then degrade the cristae of the neuron and can lead to dementia. Hopefully this video helped give you a better understanding of how protein digestion works in our bodies and how great of an impact it can affect our whole system.